Good evening, everybody, and welcome to our, Good evening, ladies and gentlemen. My name is Tanya Reinhardt, and on behalf of the University of KwaZulu-Natal's College of Agriculture, Engineering and Science, I would like to welcome you to our first online talk this year in celebration of National Science Week. Before I introduce our guest speaker for tonight, just some house rules. So please make sure that you have muted your microphone, bottom left of your screen, crossed out microphone. So if it's not crossed out, we're going to hear you. Please make sure that it's switched off. Please also switch off your camera to just to save some bandwidth. We will also have a question and answer session after the talk. You can also post questions in the chat. So just a little bit of background about National Science Week. So this is an annual celebration of science, engineering and technology, and this attracts thousands of learners and members of the public to workshops, science shows and lectures, which are held at universities, schools, science centers and public facilities countrywide. The Department of Science and Innovation leads the initiative, and the South African Agency for Science and Technology Advancement, NRF SASTA, is responsible for its implementation. Each year there's a different theme, and the activities usually revolve around this theme. This year's theme is Transforming Lives Through the Evidence-Based Science. The theme will expose basically the public's to evidence-based approach of problem solving and decision-making, responding to the situation by using the available information that has been researched or experienced. It will inform the development of National Science Week activities across the diverse sectors and allow a robust engagement among the publics. It will entrench the concepts of science as a systematic study of structures and behaviors of the physical and natural world through observation, experimentation, and the testing of theories against the evidence obtained. And tonight we are celebrating this special week with a very young researcher, and it's an honor to have her here, Dr. Afrah Khairallah. Dr. Khairallah, and I practiced this beforehand, Afrah, so please accept my apologies. Dr. Khairallah is a postdoctoral research fellow at the Africa Health Research Institute. Using her specialty in bioinformatics, she delves deeper into bioinformatics and data-driven biology for HIV/AIDS vaccine immunology and immunogen discovery research at Ivory. She's a bioinformatician, a computational biologist, and with a diverse background in biotechnology, molecular biology, comparative genomics, biophysics, and cheminformatics. So you can see she really knows her stuff. Her fascination with science began at an early age, growing up in a small town, and I'm dying to hear which town it was. She was always eager to explore the natural world and find answers to the questions that filled her inquisitive mind. With an unwavering passion for unraveling the mysteries of life through data analysis, Dr. Khairallah pursues a bachelor degree in biotechnology with a focus on bioinformatics. After that, she earned a fellowship from the Organization for Women in Science for the Developing World, OWSD, to pursue her MSc and PhD at Rhodes University. So recognizing the gender disparity in STEM fields, Dr. Afrah, I think that's how they usually call her, passionately advocates promoting diversity and inclusivity in scientific research. She supports and mentors aspiring female scientists. Afrah's research aims to shape the landscape of bioinformatics and HIV research and pave the way for future generations of bioinformaticians. So, and I know her because she's currently running, together with colleagues at the Africa Health and Research Institute, a learner program, which we are very happy that they are hosting us there at Ivory. Okay, I'll leave the stage, enough of talking. I'll leave the stage to you, Afrah, and please, if you can unmute yourself and show your face.
Right, thank you very much. That was a great introduction. I do appreciate it. And I'll share my slides. First, I would like to appreciate this opportunity to give this public lecture, and for the sake of time, I can start. So I hope that my slides are visible and I'm audible. If someone could confirm for me.
Yes,
yes,
all good.
perfectly.
Alrighty. So I also had something to share regarding the National Science Week. So I would first like to start by sharing my words. Today we gather here virtually to celebrate the essence of human curiosity and exploration during this week. This week marks an important occasion where we pay tribute to science and the achievement of science, also the pursuit of knowledge. Science as we know it is the bedrock of progress and innovation. It empowers us to unlock the mysteries of the universe, to unravel the complexities of life, and to harness the forces of nature for the betterment of humanity. Throughout history, scientific discoveries have paved the way for advancement in medicine, technology, space exploration, and so much more. We stand here today on the shoulders of giants who dared to question and dared to dream and dared to seek answers beyond the horizon of the known. Climate change, resource depletion, and global health crisis remind us of the pressing need for scientific solutions. So during this week, let us embrace the wonder of science. Let us recognize the key role of science that it plays in shaping the future and fostering innovation and catering a more sustainable and prosperous world for everyone. As we celebrate this week, let us pledge to support and encourage the pursuit of science in our communities, schools, and institutions. And let us inspire young minds to pursue careers in science, technology, engineering, and mathematics, and kindle the flames of curiosity that would lead to tomorrow's breakthroughs. Together, let us honor the spirit of exploration and the pursuit of truth that science embodies. Let us cherish this week as a reminder of the boundless possibilities that lie ahead. With that, I would like to start my lecture, and may our passion for science continue to shine brightly and guide us toward a future with limitless potential. So my name is Afrah Khairallah. Today I'll present to you my talk regarding the bioinformatics in HIV research. I'll start with my first slides. Moving to the next. So during the next few minutes, I'll take you through a brief introduction about myself, and then also another introduction regarding science, technology, engineering, and mathematics. I'm also going to introduce the field of bioinformatics, as well as the field itself in my current research at the Africa Health Research Institute. So first, let's start with the introduction and you getting to know me. My name is Afrah. I'm a researcher, I call myself now. I'm an ambitious individual and driven. I'm always up to a challenge. I'm committed, self-motivated, and determined. Outside the lab, I'm just a normal human being. I enjoy outdoors and nature. I enjoy meeting with people for intellectual conversations. I also enjoy hiking and music, as well as dancing, though I'm a terrible dancer. So I was born in Sudan, 45 minutes away from the capital, which is Khartoum. So the city is Wad Madani. And then at the age of 4, we moved to Saudi Arabia, where I grew up in Jeddah. The second born, we were 6, so I have other 5 siblings. And then I completed my high school studies in Jeddah. And then after that, I moved to South Africa for my undergraduate studies at the University of Pretoria in Gauteng. and then moving to Rhodes University for my master's and PhD degrees, and then my last stop here at Africa Health Research Institute. My educational background is I had a Bachelor of Science Biotechnology, followed by an MSc, a BSc Honors in Bioinformatics, as well as MSc in Bioinformatics PhD. So you can tell that how much I'm passionate about this field, that I chose it till the end of my studies. I then moved to Ivory, where I joined Prof. Tumbi Ndungu's lab, and for a postdoctoral fellowship in the year of 2021, December. And you can see everyone is smiling, having a great smile. It is a positive environment to grow and thrive, and using our leader as an example for that. So when I joined the lab, or joined ARI, let's say, I've realized that there is more to science than actual work of it. I've realized there is science awareness and science communication and science education, and they are different and important, as important. So I dived more, and I decided to come out of the lab for a little bit and not just focus particularly in just science, my research, so that I can engage with people and communicate my science and see what I get in return. So it was quite rewarding, and it's something that I would like to continue doing. This was the National Science Week last year, 2022, where we had the lab tours. So the first, I was engaging with the learners here at ARI, and then also a radio station project, as well as judging during science fairs and more. Also here, I visit local schools and try my best to engage and educate and create awareness for science. And I found out that this is something that I really like, and I would also continue doing beside my actual research. So that's about my journey. So I wanted to brief you about science, technology, engineering, and mathematics, and why that I'm mentioning that, and why is it so important. So as we know, we are witnessing the fourth industrial revolution. We have gone so far through four, or witnessing, let's say three, and we are witnessing the fourth. So the first revolution was in the 1765, where we had the coal revolution, followed by gas and oil, which was another revolution that changed life, altered it completely. And then electronic and nuclear, and then lastly, the internet, apologies. Lastly, the internet and renewable energy. So this is the revolution that we are witnessing now, and let's see how we could use that for our research. If you look at this, we are progressing, and some people would rather refer to it as natural progression, but we are returning back to our core. We started like about 3 million years ago, and we went through these revolutions, the coal, the gas, the electronics, and lastly, the internet. And then we are witnessing, or we're going to, well, the prediction is that the fifth revolution would actually value the human-centeredness, sustainability, as well as the environment. So this is the fifth and beyond, which will create a better world for everyone through discovery, through innovation, that will create a sustainable environment for everyone. So that's how we are seeing it, but we cannot predict, but rather utilize what we have now, which is the fourth revolution. So with that, I want to speak to you about high performance computing. And we usually had one processor, if not two, in a laptop. So I want to introduce to you the supercomputers. If you look at these are high performance computers, very large, very big. They host a big number of nodes. We call them nodes, and each node will have a number of processors. So all these computers are compacted together into one big supercomputer. And this supercomputer is so powerful that it can solve many biological or astronomical problem, or could also be applied to industry. So these are powerful computers. Now we need to know what we could do with such power, or how can we use it for the best. I want you to pay more attention to the next slide. This is one of the supercomputers placed in Barcelona, Spain, in a chapel, actually dated to the year 1940. And you can see the the supercomputer, which composed with many nodes and CPUs, or we call them also cores, is there maintained in this chapel. So this is I find it very artistic. So it's not placed in a lab, rather in a chapel. So now that you have the power of the computer, how does it work? So instead of one processor working at the time, it relies on a concept that is called parallel computing. So all the processes work at the same time. So if you have one huge problem, let's say you have a biological problem or a function or a program that you want to run. So instead of this being run on one CPU that it processes, the whole problem is broken down into pieces, and each of the CPUs takes part of the problem to be solved. So instead of doing your analysis or your simulations or whatever you are doing in the cluster, instead of spending days, sometimes it could be months actually, it could take you a few hours to complete your task. So that's something that we really need to use and apply to our research and change the way we conduct our research from traditional to a new way of doing it. So you're probably wondering if South Africa is taking part or doing something, or actually being part of this supercomputers race. I want to introduce the top 500 list. This is a list of the supercomputers internationally that has been ranked. They rank it every year in a big conference, the ICES conference. And then the fastest computer in Africa now stands or sits in Morocco. And this is just a photo of it, Tobacco. So this is the fastest so far. South Africa also had a supercomputer, which is the Lengau. It's Swana for cheetah. And the Lengau petascale system, during launch, or when they launched it, it was in the year 2007, and it was ranked 121 within the 500 top list. So South Africa was part of the top 500 list in that particular year, but then now they are out of the list. However, there's more promising news. This is a collaboration of the CSIR, Department of Science and Innovation, and also NICIS, they call it the infrastructure. So let me show you the next project, which is, before moving to the next project, I'm sure you're curious about the fastest computer up to date, actually. So the fastest computer now sits in the USA. It's called the Frontier supercomputer, and it blows out of the water all of its encounters, even its nearest competitor that sits in Japan. So USA takes the first place, followed by Japan for the most powerful supercomputer. South Africa is part of the Square Kilometer Array project that aims to build the largest radio telescope. So this would be, it will introduce 2 supercomputers. This actually was released recently. And then the next 2 supercomputers will be entering the top 500 list maybe in the next few years. So South Africa is coming back to this race of supercomputers. But then there's no point of having those supercomputers if we cannot utilize them and apply them to our research, in my case. But before moving to my last slides, I would like to also introduce the quantum computers. And this is another game changer in which the previous ones, they are classical computers based on 1 and 0 bits. But then the bits are the way of crunching or processing. It's either 1 and 0 to process your data. However, in a quantum computer based on the quantum phenomena, the 1 and 0 states could occur at the same time, which is called the superposition. This phenomena allows the computer to now become even more, adding a third state. So instead of having 1 and 0 as a state, you have a third state where 1 and 0 occur at the same time. And with that, you have even more powerful computer. And that's the power of quantum computers. However, they still have some drawbacks, but scientists are working on it. And these are mostly engineers in terms of temperature. If it increases the temperature, there will be errors while you're doing your calculations, and also writing algorithms that utilizes the new qubit instead of the bit, the classical computers. So it's like starting all over again with the classical computers, but now using a quantum computer. So the future is promising, and what we can do is just imagine. And with all the power and the computational power that we have, if we actually use it in the right way, we might actually have much better outcome. But then I want to quickly address the gender gap in STEM that actually remains significant, with about only 28 of the STEM workforce women actually make. And this is quite problematic because these environments are not created to accommodate the women in science. So we would like to address this gap and find a solution. But first of all, we need to be aware that there is a big gap, and that we need to bridge, especially in those fields. And these fields are actually one of the highest paying fields, and the women are not actually representing, they do not have an equal representation. As you can see in the biological science, it looks quite fair, but as you move down to chemistry, physics, engineering, the rate drops significantly. So that's something we really need to address and look to solve in future. So the second part, as I mentioned earlier in my outline, would be the bioinformatics itself. Let me just quickly define it. What is it? What is bioinformatics? There are definitely many definitions to it, but we can just stay with a simple one. That is an interdisciplinary field of science that aims to develop methods, software tools to address biological data. That's the simplest we can put it. Many fields together that can allow us to address our biological data. However, bioinformatics is like an intersection of all these fields: statistics, biology, biochemistry, computer science, engineering, mathematics. So it's like the intersection in the middle. Some people would like to refer to bioinformaticians as like unicorns, because you find someone who's interested in all these fields at the same time is quite rare. You find people developing passion for a certain field, but for someone who want to bring everything together, that somehow makes it a bit rare. So you're probably wondering, what do I need to do or to study, or what skills do I need to actually enter this field? So you definitely need some statistical and mathematical skills. You need some strong programming skills. General biology, you can do with that, or you can have more. If you have more knowledge of genomics and genetics, that's actually a plus. Database management is also a skill that is required for the field. And the very last one is data mining and machine learning, which is a plus actually now. But if you have it, it will be a great addition to your skills that allows you to take things further. So that is bioinformatics. There are different fields in bioinformatics. We have disciplines. We got the sequence analysis, the software development. So the bioinformaticians divide within this field, they divide even further. You will find a structural bioinformatician. You will find someone, bioinformatics that deals with genes and genetics or genomics, transcriptomics. You get drug design and development. I labeled in the same color because it's more linked to the structural bioinformatics. You get network and system biology field. So all of these fields are supported by the same, or within the same umbrella. And to have a supportive system to establish a bioinformatic core, you would need a database management, you need a strong infrastructure of computational resources, you need your engineer to assemble these and make the most out of it, write scripts that allows these big supercomputers to communicate efficiently for faster calculation or faster output. So that's all what you need to establish a core or harness it the most. So I would like you just to refresh your memories. I'm sure maybe you have, but this is how a DNA look under the electron microscopy, just like a thread. And in this figure, they're showing us that the DNA is unwinding. If you could, there's a little bit of opening. So that's how a DNA looks under the microscope. I just wanted to bring you back to the biology, now that I've mentioned a lot of computational work. So now we are looking at a DNA, and that's exactly how it looks under the microscope. So what we exactly do, how do we link the two, the computational and the biology? You start by basically getting the samples, drawing the samples from whatever cohort of patients you're studying. You can isolate the DNA or you can isolate the mRNA. Given that this is the central dogma, we have our DNA transcribed into an RNA, translated into amino acids that makes a polypeptide. That polypeptide come together, assembled into a 3D structure folded protein. So bioinformatics comes in every single step of these. You find it at the genomics level, at the transcriptomic level, you find it at the proteomics level, and lastly, at the protein level or the structural level. So that's, you find your bioinformatician in all of these areas. So this is, I wanted to link it to biology. So the data that comes is different in each. Let's talk about drug repurposing or drug discovery. So now this is a pipeline that shows you how many years it takes to actually come up with a drug. From basic study, designing, experimental, you do in vivo, in vitro as well, and then you move it to the different phases, and then you take it to the registration at the end. So what I propose is, why not accelerate this process? Why not use the resources that we have and accelerate this process, and also repurpose the drugs that we already have to speed up? So this drugs will be registered, FDA approved. So what you need is to perform screening. The screening does not have to be in the lab. You can actually do it in a computer, given that you have access to a supercomputer. You perform your screening and you take it to the next level. So that's actually one of the bioinformatics part, which is the drug discovery and designing. There's also part for natural product as well, where you can have a natural product and you also screen it in silico. So what we say about this is an in silico screening. So that's a new term that we add into this pipeline. So it can accelerate the process, and that's something good. This brings me to my PhD research, and I like to refer to it as targeting a killer. I was working on malaria, and I was also working on drug discovery project. So this is the pathway that occurs in the parasite or the malaria parasite. And this pathway, it's important for the parasite cell division and growth. So our aim was to inhibit this pathway or blocking it. We do not want it to occur so that the parasite does not survive and then get cleared from the whole system. This pathway has been targeted before by antimalarial. You know, these are the two drugs the two targets, enzymes that were targeted by common antimalarial, let's say artemisinin and pyrimethamine. These are antimalaria drugs. So they targeted three enzymes in this pathway. So my project was to look for alternatives. And I actually looked at the first two enzymes, the GCH1, the GTP cyclohydrolase, and the PTPase as well following. So these are the enzymes that I was targeting and trying to inhibit, but I was not doing it in the lab. In fact, I was doing it in a computer and using the supercomputer. So I conducted sequence analysis. I had the structure of the enzyme, so I performed structure analysis as well to see how the structure of the enzyme, how it moves, and I'll show you how just now. And these are compounds that binds to the enzyme, the targeted enzyme. So we call this a computer-aided drug discovery approach. It integrates all these steps, from the sequence analysis to the simulation, to the screening of compounds. And these compounds that I was screening, they were natural compounds, and I could also repurpose, but that was the aim of the study. So if you want to look at it, studying the structure itself, I'm not sure if I can have it moving. You can see this is the dynamics of the enzyme that was modeled, and it's as accurate because we are following the Newton's the first law of motion. So these are all calculations that has been done on every single residue. And then we use the supercomputer so that it can do it for 1,000. This enzyme had 1,600 residues, and then it accelerates the movement. So it predicts the movement of this enzyme. In any way, we call it a simulation. And then we found out that the tunnel of this enzyme is what is important, given that it shows high mobility. This is another part where we did, I did the screening. These are the compounds bound to the enzyme itself, blocking its tunnel, which is important as shown before, with the highest mobility. And the simulation shows the compound staying in the center of the tunnel and not moving out and binding with a very great affinity. So that was the study itself. And from there, we proposed this compound as one of the head compounds. That was my PhD, and it could be nice if we can move it to the next stage where we tested in vitro and also in vivo and take it further, and maybe we could come up with a drug, but that was for the PhD work. And lastly, I want to mention my work in HIV here at Ivory. So I try to transfer my skills in bioinformatics also to another field. Instead of the structural and the discovery, now it's into the HIV research. The main aim was to find or to the pursuit of an effective HIV vaccine. So the challenge for still remains in HIV to develop an HIV vaccine, given that the virus itself mutates at a very high rate, so it escapes the immune system. It's like it has a different shape every time, and the immune system could not recognize it. So that's why the challenge lies, and that's why we took longer to come up with something as scientists. So, however, we can say that there's a lot of studies that has been done to understand the human immune response to it, as well as the complexity, the HIV interaction as well with in the humans. So all these studies somehow paved the way for rational vaccine design and also optimized vaccine strategies. So scientists are all coming together for finding a novel way of a vaccine. So what we aim here, well, in my department, in my lab, is to develop and design a vaccine that emphasizes a certain cellular immune response. And that response, in particular, if you want to refer to it, is that we want to find an immunogen that is capable of introducing certain antibodies. We call them broadly neutralizing antibodies. So we want an immunogen that produces more of these antibodies. I'll explain this as I go further. But before going so, I would like this presentation to be as public as it can. So I want to always bring us back to the initial definition. Where do cells come from? They differentiate from our bone marrow. You get different type of cells that have different functions. But then in our study, we are focusing on the B cell. You probably ask why. The B cells are the one responsible for releasing antibodies, and antibodies are important for us to fight diseases. And then they neutralize whatever antigen. But when I said antigen, I mean a foreign object. It could be a virus, could be a bacteria, it could be anything. So it's a foreign object to your body, and these are the antibodies released from the B cell to neutralize or mark this antigen to its death, so it doesn't bind to your cell. So that's why. If we move to the next part, and that's where the study actually starts, there's a cohort, a fresh cohort, female rising through education, support and health, the fresh cohort. We have about 15 samples, but we are aiming to add more. And before that, I would like to have a photo of my colleague. We are working in collaboration, and this is purely her work. 15 samples and hoping to add more, divided into 3 main categories. We have healthy individuals, we have people who just received the treatment and then being followed after a year for a long term. So we call it pre-treatment and post-treatment and healthy. That sounds like a good logic. We would like to compare all the B cells in these individuals to see what's going on. So what she does now in the lab is isolate those B cells from all these individuals. We isolate the B cells. We are interested in the antibodies. We need to know the landscape of the antibodies that are in these individuals. What is it? What is the collection of the antibodies, given that it's different from a healthy individual? It's actually different from one healthy to another healthy individual. So we're trying to understand the landscape of the B cells first before we move to the immunogen. And for that, I work closely with my colleague, Dr. Judy Magura, to do that. I get the raw data and she does the wet lab, and we work in collaboration, and it seems like it is flourishing. So what we are aiming to do is to jumpstart the immune system. How do you want to jumpstart it? We have the B cells, as I mentioned. a collection of B cells in our body, but then we are looking for a particular B cells. These B cells are the ones that are able to produce antibodies that neutralize the HIV virus. However, the frequency of these B cells is not the same. So you can see that there's more yellow than blue and more, and I would refer to this as red. So the structure or the landscape is different. So that's why the first aim of our study is to see what's going on, how many, the frequency of the B cells, how many reds do we have, how many greens do we have in healthy and not healthy, people under treatment. So that's the first aim. Then the second aim is to prime. Priming is the method that we use to induce certain B cells. So we will use an immunogen that gives us more red. And that's how we jumpstart. So you have little red here, you prime, you get more, and then you boost again with another immunogen that gives you a bigger number of the particular B cells that you're looking at. And with that, we can maybe now have a vaccine. And we're using a lot of techniques to achieve that. So that's the main approach, if I could explain it clearer. So what happened is we have the B cells, it has the antibody, and this is just an enlarged image of the antibody. What we sequence is this region that has been shown here. We don't sequence the whole antibody, it's just this particular region that shows the difference among the individuals. Everything else is constant. This is the B cell, now it's been sequenced, and then we need, in another way, we're decoding this antibody or B cell into its genetic material. So the decoding gives me a number of short reads, bases. It comes in a text file, raw file. So that's where my work starts. Now I have small fragmented reads. The first step is to do quality control. And then after that, making sure that these reads are actually accurate and it's not due to coming out of error. And then assemble them together, perform an annotation. Annotation is when we map it against like a common biological databases, which are available online. And then after that, we do the comparison. I will try to break this even into simpler words. So I'll repeat myself in the next slide. This is how it looks like. So the antibody that has been isolated by my colleagues and sequenced and decoded, it comes to me in a text file that has bases. What you see here, these are the different bioinformatics file formats. You have a FASTA file, a FASTQ, you get, and these are all abbreviations that stands for something. FAST-all, it was called FAST-all for all protein nucleotides. It used to be FAST-P, FAST-N, and that, but they changed it to FAST-all. So you can get a sequence of amino acids, not just bases, DNA, like A, T, C, G, no. And then you can get FASTQ that shows the quality of each bases. You can get a single alignment format or a binary alignment format and BCF variant calling feature format. So these are all the formats that different bioinformaticians work with. In the field of genomics, you work with these text files, FASTA and FASTQ, SAM and VAM if you're performing alignments. So that's how it looks like. Then what do I do with it? If you look at this last figure here, it shows you everything comes to me fragmented and I need to make sense out of it. I need to assemble it, put it all together, and then find the meaning, what's in it, what is the biological meaning of it. So if you look at this last sentence here, that says nothing in life is to be feared, it is only to be understood. Now is the time to understand more so that we may fear less. Now this comes to me fragmented. I do not know the message. I'm lost and I'm looking for a message. Then I get those fragmented reads. And then you as a bioinformatician, then you try to encode or decode. You look for these fragments and try to map them back to the reference. And what we are mapping against, these are biological databases. So we take our reads and then we assemble them until we get a full strand of sequence. And those full sequence stand, and then we compare them to each other and see what is the difference. Is this mutation there? If it's not in the healthy individual, what is the landscape, as I mentioned earlier? So that's how we do it. The very last part, this is just some real results now that we're somehow excited about, but we still need to polish it even further we are looking at the frequency. I did not actually explain earlier that we have within the antibody itself, we get different families that occur at different frequencies. So as the first step is knowing the structure, what's in it, the frequencies. We're just looking at the first, we find that this family in the antibodies that we are studying occur with the highest frequency, followed by another region and a family within another region. So these are the initial result that we are still yet to polish. And then also visualization. This is plotted in a scatter plot, and then you can also just plot it in another way in a heat map format, and what you see in red is your highest. So let us conclude, and then after that, I'll open the stage for questions. So the field of bioinformatics. I would like to once again reinstate and say it's the acquisition of knowledge by means of computational tools. So if you are interested in computing or coding, why not gain a bit of biological knowledge and then try to combine the two together and get the most out of it? It allows you to organize, manage, and mine your data, and we need more. This is the field is growing, and we require more of the bioinformatician. In fact, there is a huge gap, even in Africa as well. So we aim for capacity building. HPC, as I mentioned earlier, the high performance computing in the fourth industrial revolution, it can accelerate our research and change the way we conduct our research. So why not use it now to get the most out of what we have and make the most of it? So what we are doing now is actually trying to combine this field, start by building the infrastructure and the tools, the capacity, so that we can even take it further. So that's with that. My very last note on the STEM, and then as I said earlier, I would like to address the gap and make this environment more inclusive, not just equal representation that we are looking for. We are looking for inclusivity, not just having the right number, but make sure that these women, when they enter these fields, they stay and they contribute, and not just just being a number in the statistics. And with that, I would like to thank you. And I was not timing myself. Maybe I did take my time, or maybe I'm not. So thank you very much for attending and listening to my talk. And with that, I can open the stage for questions. Thank you very much.
Thank you, Afrah. And you are, yeah, you've flew through it. Fantastic. And we've got some time for questions. Thank you very much for this really exciting talk and really interesting things. And I usually I'll take always the liberty as being a host to take, to ask the first question. And I have to say, I don't do biology, so. You were talking about the mutations, the immense mutations of the HIV virus.
Okay.
Is it, if you would have unlimited computing capabilities, so no restrictions whatsoever, would it actually ever be possible to predict mutations or not?
Beautiful. Yes. My answer is yes. You can. So what you need is to model, and this you just answered yourself. My question to this is yes, it's a brilliant idea, and so far no one has dived into this, but you can predict mutation, and that would be a novel study if we can predict the mutation of HIV. I believe that we have enough data or information that's made available, but we just need to start working on it and predict the future mutations so that we can be one step, if not more steps ahead of the virus with the power that we have, the computing power. So this is a very, very good point. Yes, we can.
Okay, I'm glad. So maybe we're inspiring somebody to, follow up on the research.
Exactly, yes.
There is another question from Rachel Nell. Rachel Nell, do you want to unmute yourself and ask the question yourself?
Sure, that's fine. I actually have two questions. but I'll start with the one I actually put in the chat. I just wanted to know, so I'm doing some biomatics, self-taught bioinformatics, dare I say, for my master's thesis. And I was just wondering if you think it's necessary to do an additional QC step after I've done my assembly.
Okay. And the second question, or should I?
And then my second question was, I used the spades binaries to do my assemblies. And one of the output files is a scaffold graph that's a GFA file. And I don't really know how I can possibly visualize that. I thought maybe I would pick your brain and see if you knew.
Okay, so I'm wondering, why is the assembly done just, the quality control done after the assembly? So the first step, when you get your reads, you filter through them, having a cutoff threshold for quality. So whatever you have later is your scaffolding in high quality. So I'm not sure why we're doing the quality control after the assembly.
Oh, no,
So,
I just wondered, my supervisor asked me if I think it's necessary, and I wasn't really sure what the answer was.
oh, okay, no, it is not. So the first step is you do your quality control of the raw reads that you get, and then after that you assemble them into contigs actually, and then those contigs, they come together into bigger scaffolds, right?
Yeah.
That's what then how do you do the quality control is by mapping them to a reference that you have. So if you have a reference genome, if you do, you can map them, and with that you can check the quality. So it's not the reads quality, it's the quality of the assembly. So mapping it back to a reference genome. So that's the first, yes. And there are many tools. You can write your own, or there are many tools that I'm not sure which one to use, but Galaxy kind of put everything together, make it automated, and it's user friendly, but we try to avoid that as bioinformaticians. So we try to avoid user-friendly softwares. Yes, and the second question? I just couldn't.
Sorry, I didn't hear that.
The second question, I'm actually going through the chat to see the.
Oh, my second question.
Yes.
Oh, I did a spades binaries assembly of my genome, and I got an output of a GFA file, and I don't know how I can visualize it, and I thought maybe you would know.
Oh, the visualization of your tool. I can't think of a tool now to visualize it because I haven't done that. But what I used before, it was Galaxy to do that. And then with Galaxy, it's automated. You cannot like it's interrupted somehow. But like we can stay in touch, and I look for a tool that we can use, the GF file, right?
Okay, awesome. That makes sense. Thanks.
Yeah, interesting question. Thank you.
Sally has a question. Sally?
Yes, I have a question. Afrah, I'm not a scientist. So my question is, you were talking about how you assemble your reads, and you had that sentence from Marie Curie, and you illustrated it very nicely. Mm-hmm. So I was thinking, you obviously using supercomputers. What is the role of intuition in making breakthroughs?
You mean the role of accelerating this process?
Yeah, your own personal intuition.
Okay.
Your work.
Yes. We have actually tried this. So let's say you have this number of reads. It allows us to do multiplexing. If you run this in a code in a normal computer that is not a supercomputer, you will have to make sure this computer is functioning for the next 2 days running through. What we do now with the supercomputer, now every bit of this reads are divided into, like, filtered, or maybe it's solved in a sequential order and then divided across the CPUs. So instead of this 2 days period where you have to wait for your assembly to be finished, you just get it done in 1 or 2 hours. And that gives you the leverage or the opportunity to add more. So if this reads, if it's one participant took you 1 week, you use the supercomputer, and then it took 2 hours. So why not still do it over 2 days, but instead of one participant or one reads that's coming from one individual, let's just now accelerate and add more. Instead of 4, we make 10. Instead of 10, we make 20. And all these reads come together into difference. So it's a reduction of time and the complexity of the problem. This also goes for the simulation. Before, the proteins that we used to study, they were like polypeptides, small proteins. Now you can study large system. You can even study a capsid or an envelope, the whole envelope of the HIV. Before, you could not do that. Your laptop maybe need to run indefinitely, and then after that it might break because you consume the memory at least. So it allows us to add more, or actually add more samples to the study. That's one, and get it done in less time. So that's what I see, and that's where we can actually now think more and invent and come with like more solutions. So this brings us to the topic of personalized medicine, where it's now possible for everyone. Instead of fewer number of people with all this computing power, we can just get it done for everyone instead of one person, and then have a drug, for example, that is not a one size fits all, but we can get a drug that is tailored for someone based on your genomics material. So there is more to it, I see.
Okay, thanks. Yeah, my interest is maybe general with scientific breakthrough. How much is it the computer, or how much is it the role of the human brain in the aha moment?
Okay, yeah, this is a good question. It takes a while to develop something, so you would like work hard. The human, I would say maybe 80, because you just need to build this machine, make it communicate efficiently, make it do proper calculation, and after that you sit back and enjoy the ride. So I feel like the human's still playing an important role. That's one. And you are still need the humans, like someone to do the quality control. Like let's say you run something, the machine will just give you raw reads. You as the scientist or the bioinformatician, you need to sit down with this output and make draw a conclusion. You cannot just, the machine, what it does for you is just the pure calculations for you, solving these problems. But then what to do with this actually is based completely on the humans. So I don't think we will ever dismiss the role of the humans. As I mentioned in that graph, we're going back to our core, where we actually center the human. It's more human-centered. So it does not dismiss the role of humans. Just like the ATMs, when they first introduced, the bankers thought that they would lose their jobs, and because now the ATM does their job for them. But now we have more banks than even before. So it was not the end of it. So that's one example that I always like to mention. I hope that answered your question. So
Yes, it did. Thank you. Just checking if Tanya's back. I got a message from her. She was booted out. Tanya, are you here?
Yeah, the Wi-Fi didn't like me, so they kicked me out. But I'm back. And James Sun has got another question. James, if you want to unmute yourself.
Yes. Thanks, Afrah, for that beautiful talk. I just have 2 quick questions. One is your sample size. You did mention that you're working with 15 samples. And at the beginning of your talk, you did highlight how HIV mutates so fast, and every time you're trying to target one thing, it becomes something else. And I'm just trying to imagine how you are accounting for that. You also didn't provide any highlights on the subtypes or distribution of your 15 samples. Are they all the same subtype like C, or are they a number of subtypes, to try and see how different things are between these subtypes? And then I'm also curious, are you going to sample at one time point or multiple time points? Because in that case, then you would be wondering, how are you accounting for the B cell maturation process? So for example, if you have at the time of sampling, certain B cells, which may later be very important, are still naive or way back in the maturation stage, like how would you account for that later on if you only sample at one time point?
Okay,
Yeah,
thank you very much, James. These are, I'll try to remember most of these. So first of all, it's a pilot study. Luckily I have my colleague Judy here as well, so we always like to tackle things together. It's a pilot study. We are hoping to add more. That's one. Talking about the mutation, yes. So what we are looking for, those B cells that are precursors for BNAbs. A BNAb now, we know that this BNAbs has gone through all these different mutations for it to develop. So we are already one step ahead of, if not many. So these BNAbs have gone through all possible mutations of the virus. So what we are looking for is the immunogen that produce those B cells for this BNAb. For someone who does not develop these broadly neutralizing antibodies, they might not have enough of these. So our mutation, we account for the mutation, but then we are looking for these immunogens that produce this broadly neutralizing antibodies that neutralize most of the different variants of the HIV. So it's not completely all of it. So that's the first part. And then, yes, it's a different subtypes, yes, to the subtypes as well is accounted for. And it's the different time points. So we follow the same individual. We have our negative control, like we call it the healthy individuals. And then we have two time points, post and pre-treatment. So that's the time points that we have so far. And we can still like polish the study further with questions like yours. So I'll make note, and I'm hoping for more discussion as well after this. So I hope I answered all your questions.
Yeah, you did. Thanks. Thanks.
Thanks, Sam.
Thank you so much. And I think our time is almost up. Sally, are we having a poll today? Sally is not with us. Ah, here we go. Do you, if you would please just help us, filling in this one here. It's just a poll for us to see. Yes, and to help us, to judge if we're going the right way and so on. Okay, and in the meantime, I just want to read out some vote of thanks. And just to remind you that we have actually other exciting talks lined up, and the next lecture to celebrate National Science Week will be tomorrow at the same time. So this year we are actually celebrating, I don't know if you know this, celebrating the International Year of Millets. And just like I don't know what millets are. So if you want to find out what millets are, or if you know what millets are and you want to find out more, we have a speaker lined up tomorrow. This is Professor Hussein Shimeles, and he will present a talk with the title Millets in 2023, Demand-Led Improvement and Community of Practice in Africa. And now I would like to take this opportunity to thank a few people.
It seems like Tanya is frozen. Has she disappeared? Okay, I think we've lost Tanya, but I'd like to thank Afrah for that very interesting talk. And thank Ari, Roanne and Hannah for sharing your expert researchers with us. Thanks to the college for hosting this for National Science Week, and to the audience for coming to listen, and to Tanya for hosting. And I think on that note, we will bring tonight to an end. So thanks everyone, and thanks Afrah.
Thank you. Bye, everyone.